All right, whew, it's bright up here. All right, well, thanks, guys. I'm Dr. Christy Williamson, and today we're going to talk about diet and genomics. So the point of what we're going to do, and we'll get through these faculty disclosures here. As Dana mentioned, I own these companies. This is what I do at those companies. But we really want to talk about the diets that everybody already knows, or should mostly know. And we're going to take a nutritional genomics approach to these diets and really think about when maybe they might be indicated and maybe when they might be contraindicated. So, and then how we take that information that we learn through nutritional genomics and craft some new information and how we actually apply that in clinical practice. So that medical nutrition intervention is really important. So that's really what we're trying to drive home here today for you guys. So since it's almost lunchtime, I'm going to torture you guys with lots of beautiful pictures of food. So hopefully I'll hear lots of tummy grumbling out there. So we're going to take a trip around the world and look at these different diets. So we're going to look at the Mediterranean diet. We're going to look at the traditional Okinawan diet and modern Okinawan diet, the DASH diet, the paleo diet, our standard American diet or saturated fat diet, and then lastly the ketogenic diet. So we're going to start with our Mediterranean diet, which has been very well studied, but we're really looking at a high intake of vegetables, legumes, unrefined grains, and cereals. And this is not the, hey doc, I like to eat a lot of macaroni and cheese diet, because I know a lot of you will probably get that from your patients or clients. Really high in fruits and nuts and olive oil, moderate intake of alcohol, primarily wine, again, not the macaroni and cheese and bottle of wine diet. Some fish, some poultry, and then low intake of saturated fats, meats, dairy, and sweets. This diet has been studied till the end of the earth. I'm sure lots of people in this room have looked at studies on the Mediterranean diet. It's generally safe for everybody when we look at these large population studies. So in a meta-analysis of 1.5 million adults, if you guys have that kind of turnout for your studies, I'm impressed. If I could get that kind of turnout for Zebra Diagnostics, I'd be really happy. So you guys can send them over, that'd be amazing. But when we look at this study, when we look at this turnout, we find a decreased risk of cardiovascular disease and overall mortality, decreased risk of cancer, Parkinson's disease, Alzheimer's disease. This is just from dietary intervention alone, okay? So again, I'm talking to a room of mostly nutritionists. You guys already know this, right? Nod your heads, yes, okay, not news. All right, so let's drive the point home just a little bit more. 15-year study looking at 10,000 healthy women found adherence to this diet, found increased odds of healthy aging by almost 50%. Healthy aging, you ask? What does that mean? It means being 70 years or older and not having any impairment or physical impairment or disease, okay? Our suspected mechanisms, again, a bunch of nutritionists and MDs in this room. We're looking at decreased oxidative stress. We're looking at decreased inflammation. We're looking at improved glucose metabolism and insulin sensitivity. Okay, not rocket science. All right, you guys are going, where is the meat? When is she going to get there? Okay, so then we're also looking at other components of this diet. It's got a pretty high fish consumption. Okay, so we're looking at this aspect of the diet and saying, okay, well, this is helping our lipid profiles, less incidence of hypertension, decreased coagulation, decreased oxidation, and decreased inflammation. So let's talk about fish oil for a second. Anybody have patients that take Lovesa or high-dose fish oil? Yes? Anybody else? Yes? Does it work all the time? No. Ever heard of a fish oil responder? Maybe? Okay. Do you think there's a SNP for that? Yeah. Anybody going once, going twice? Of course there's a SNP for that. Let's learn about it, okay? It's called nitric oxide synthase 3, okay? This particular SNP determines whether taking omega-3s will decrease triglycerides. This might be pretty important when we look at all of those studies that say, oh, you should take omega-3s if you have high triglycerides, okay? So if there's a change in this gene, NOS3, that means that there's a greater likelihood of being a fish oil responder. Okay, that means that the wild type of having this gene means that you're going to be a non-responder. So sometimes if you have a SNP, that means it's a good thing. Sometimes it means it's a bad thing. Sometimes it means there's no change. All right, This time, if you have a change in the SNP, that means it might actually be a good thing, kind of, sort of. Okay, so let's look and see what the study actually said. So the subjects that had the change in the allele, they found if they had low levels of omega-3s and had a 25% increase in serum triglyceride levels, that when they took omega-3s, that they were responders. Okay, so this is a really interesting study. This is one of those things that we look at and say, okay, if we are going to prescribe or recommend, if you're a nutritionist, fish oil, then this could be pretty important when determining whether or not we're going to give this recommendation. So this begs the question, if you don't have this change in the allele, if you're a wild type carrier, does it mean that the Mediterranean diet or fish oil might not be good for that patient? Of course not. Okay, so we can't cover every single thing that has to do with fish oil regulation or the Mediterranean diet, but this is one aspect of care that you can start to look at and start to integrate into your clinical practice. One thing I want to really drive home here is that SNPs do not equal disease in every single case. Okay, so do not get in your brain that when you talk about one SNP, that it's going to equal a disease, okay? Nor does it equal optimal diet. When we do optimal diet panels, they have hundreds of SNPs, okay? We start looking at these algorithms. We start predicting all of these different things. They all interact and all play together, sometimes nicely, sometimes not so nicely. But if you're looking at this particular SNP for your patient, there are other ways to lower triglyceride levels, like a low carbohydrate diet, okay? So it doesn't mean that it's the end of the road for you. It just means you need to start digging and looking other places. All right, so some other considerations, since we can't cover, again, every aspect of every part of the diet, but think about the Mediterranean diet. It's really high in legumes and beans. So does this person have lectin sensitivity, SIBO dysbiosis? Same thing with unrefined grains and cereals, yeast overgrowth. Do they have a genetic requirement for a low carbohydrate diet? Promise we'll get to that. Fruits, fructose intolerance, again, yeast overgrowth. Really high in nuts. Anybody heard of oxalate sensitivity, genetic hyperoxaluria? Yes? No? Maybe so? We got a hand raised back there. Good job, Jan. I knew you'd know. Okay, over there. All right. So if we have some genes that are listed here, so for type 1, type 2, and type 3 of genetic hyperoxaluria, these typically will present with myalgias, often kidney stones, those kinds of things. So these are some things that you should be thinking about when you're prescribing diets or recommending diets for people. Alcohol can often be high in sulfites or sulfates. We didn't really talk about it, or Jessica didn't get a chance to talk about CBS upregulation, but so much. But this can cause sometimes a sulfur sensitivity. Again, it really needs to be validated before you start treating for that. But sulfur sensitivity can be an issue. Alcohol and fish are really high in the Mediterranean diet. Again, you might need to be thinking about histamine sensitivity. So I've listed some histamine-related SNPs here as well. So when thinking about diet in general and the intersection of nutritional genomics, it's pretty complicated, all right? There are a lot of things that you need to be thinking about if you're really going to be integrating these things into practice. But the good thing is that once you start using these things in your daily practice, it becomes second nature, okay? You start thinking about, oh, there is a SNP for that. Oh, there's a pathway that interacts for that. You begin to think about these things, and they begin to integrate pretty easily. All right, so moving on to the DASH diet. This diet is pretty simple overall. It emphasizes fruits and vegetables, low-fat dairy, and we use this to treat hypertensive patients. The Nurses' Health Study found that there was a decrease in coronary artery disease, and mostly because of the decreased intake of saturated fats. The problem that most people have with this diet is compliance. So if we look here, we have the 2,000 calorie diet over here. We've got about 7 or 8 servings of grains, 4 to 5 servings of fruits and vegetables. This little marker keeps going away here. 4 to 5 servings of fruits and vegetables, 2 to 3 servings of low-fat or non-fat dairy, very low intake of meats and fish and poultry, moderate servings of nuts, seeds, and legumes, and very limited fats and sweets. So pretty tasteless overall. So we're looking at a similar macronutrient ratio, but with the exception of much lower salt and much lower fat. Another thing that this diet does is it replaces olive oil with canola oil. So yeah, yuck. So Dr. Yael Jaffe actually published a study on nutrigenomics and inflammatory fat that looked at how our body actually processes fat differently, and she'll a speaker later on this week. And so when we look at these things, it matters what type of fat and what type of gene and maybe your gender and how all of these things interplay. These things are important, and I'm glad you guys already know that, and I got the response. So that's really what we're looking for with these kinds of things. So, you know, the DASH diet, it has its flaws. Some pretty smart people came up with a solution to the compliance factor. They started to remove some of the carbohydrate and increase the monounsaturated fats about 10%. They found that we still had cardioprotective effects, blood pressure was still down, and did not have an increase in LDL. VLDLs were still coming down with this diet. And compliance, of course, went up because the taste factor went up. Because no fat, no salt, again, cardboard, sounds delicious. All right. So next we're going to talk a little bit about the Okinawan diet, and really we talk about hypertension and salt. Again, the DASH diet we use for hypertensive individuals, but again, compliance factor is not so great. So let's look at the Okinawan diet. So overall, we have the traditional Okinawan diet and the modern Okinawan diet, but looking at the broad-based studies, 60 to 85% carbohydrates, 10 to 16% protein, and pretty broad between 16 and 26% fat, with only 2 to 7% of that being saturated fat. So again, pretty low restriction on saturated fat. Overall, very low calorie diet, high consumption of vegetables, typically purple sweet potatoes and yellow and green vegetables. Pretty high soybean consumption, often in soy sauce. Moderate consumption of fish, low protein, often in low-fat pork products. Little bit of dairy, very low saturated fat, so we have a very low omega-6 to 3 ratio. And we have a high emphasis on low glycemic index carbohydrates with a high fiber intake. Moderate alcohol consumption, mostly in sake, and then fairly high in sodium. So when we're looking at the broad base of this diet, it's generally safe for everyone. It can be high in sodium, and so this is where we start looking into perhaps being contraindicated for those who may have issues with salt sensitivity. Generally speaking, very similar in studies as the Mediterranean diet, it's found to decrease the risk of cardiovascular disease, Alzheimer's disease, and Parkinson's disease. So take a look here at sodium factor. So Mediterranean diet, and I want to focus on the modern Okinawan diet and DASH. So Mediterranean diet's close to RDA at about 2,000 milligrams. Traditional Okinawan diet and DASH diet, right around 1,000 milligrams of salt, so pretty low. Modern Okinawan diet getting up to 3,200 milligrams, so very, very high in salt. So let's take a quick summary here. DASH diet we're going to use to treat hypertension. Okinawan diet may cause hypertension. Mediterranean diet is neutral, may decrease hypertension. But hypertension is kind of a moving target, okay? So medically speaking, you go to a doctor and they're going to say, we don't really know what's causing your hypertension. You may want to go on a low salt diet, but they're not really going to know why they're telling you to do that. Okay, this is a complex disease. It's one of those medical mysteries most of the time. So one of the things that we're going to focus on genomically is a hormone called aldosterone. And aldosterone is very closely related to cortisol and stress and obesity. Okay, so we're going to take a little trip and look at some SNPs that are in this particular pathway. The first one is the angiotensin converting enzyme, or ACE gene, which was talked about by Sharon in the first speech. And this again is a deletion. When you have this deletion, it causes an upregulation of angiotensin 1 to angiotensin 2, which ultimately causes more aldosterone to be in your system. This mildly increases stroke risk, and it can cause learning and memory problems, anxiety, autoimmunity, and hypertension, and of course, stress. When we have this, we're really looking for validation. So what Dr. Pisano was focusing on for you guys was making sure you understood the validation concept. So when we have this alteration, we're going to have decreased sodium excretion. So when you have hypertension, typically you're going to have swelling, right? You're going to have edema. So this is what's happening physiologically. And you're also going to have increased excretion of potassium, so potassium wasting. So on a blood test, you would find that someone has high levels of sodium and low levels of potassium. You're also going to find that they have high levels of aldosterone, and clinically, typically, they will have hypertension. So for those that have this altered genotype, we typically recommend that they restrict sodium to about 1,500 milligrams a day. Now, when you look at the studies, we typically find that this is often found in people of Japanese, Spaniard, and American descents. If you look at those diets, we're looking at a standard American diet, which is often very high in French fries and hamburgers, so very high in sodium. The Spanish diet is traditionally very high in sodium as well, lots of cured meats. And of course, the Japanese diet, we're looking at the modern Okinawan diet, so very high in salt. So the angiotensinogen gene is in a linkage disequilibrium, which you also learned from Dr. Hausman this morning. So this is co-inherited often with the ACE deletion. So this specifically exacerbates the ACE deletion, so it increases angiotensinogen levels. This gene all on its own is associated with insulin resistance, and it can be associated with preeclampsia and cardiovascular disease as well. So one of the things that I want to talk to you guys about looking at these two genes is that they're part of the RAS system, or renin-angiotensin-aldosterone system. And this is a hormone system that regulates blood pressure and fluid balance. Now, I'm a biochemist, and I'm not going to scare you. I'm going to take the teeth out of this pathway, but we're going to look at some biochemistry next, okay? So first, I want you to look over here at the boring pathway that doesn't have any pretty pictures. But we're going to look at the conversion of angiotensin 1 to angiotensin 2, and this is regulated by ACE, okay? So if we have an upregulation, we're going to make too much angiotensin 1, okay, or angiotensin 2, excuse me. And then we're going to come down here, and we'll talk about the AT1 receptor in just a minute, but the ultimate response is that we're going to have too much aldosterone, okay? And you see here that we have end organ damage, all right? Now remember, angiotensinogen is the AGT gene, okay? So this exacerbates the system, and it's first, because being first is important, right? So it's going to basically push more issue into this pathway, okay? So that makes sense when we're looking at these issues, okay? Now, if we're over here in the pretty picture, we also have angiotensin 1 converting to angiotensin 2. And then we come over here and we have vasoconstriction. So we come down here, we turn up the pressure, we have blood pressure, okay? Over here, we have aldosterone synthesis being created from this particular pathway. We have potassium wasting from the kidney and sodium retention, okay? So here we have, in a picture, in a nutshell, what's happening. The one thing that we didn't talk about was how angiotensin 2 is going to increase our sympathetic nervous system. Anybody tell me what that does? Fight or flight, right? That makes us pretty stressed out. Anybody have any stress in their life? Everybody pretty relaxed? Feeling pretty good? Yeah, on vacation? Everything's good? All right. So let's talk about what that means in terms of what we can do clinically, okay? So we've talked about what it looks like in the lab, what it looks like physiologically in pathways. So aldosterone is over here. So if we have ACE and AGT and we naturally have more aldosterone, let's just look at that in terms of perspective, okay? So aldosterone is just hanging out over here. Acetyl-CoA, that's part of what pathway? Our citric acid cycle, or Krebs cycle if you're old like me, okay? So acetyl-CoA is way over here, and this little guy keeps going away. All right, and then we have cholesterol and LDLs, all right? So then we start thinking about cardiovascular disease, right? And then we have pregnenolone, all right, and progesterone. So this is what we call our pregnenolone steal. All right, so if you have aldosterone, and aldosterone is made from these precursors over here, and you're just busy making lots of aldosterone, aldosterone says, oh yeah, come here, pregnenolone, you beautiful thing. And it just starts stealing it and pulling it all the way over here. And it just keeps taking and taking and taking. And then aldosterone happens to be right next to cortisol, okay? So naturally, even if you don't have a drop of stress in your life, you're going to make more cortisol out of this system. If you happen to have a boss who's just not very nice to you, or you happen to have stress in your marriage, or I don't know, your kid's getting on your nerves, or whatever it happens to be, you're gonna have a little bit more cortisol. So that's gonna pull more on this system. And it's gonna start saying, okay, we need more cortisol. What happens when you have more cortisol? You have insulin resistance, right? So you already have more aldosterone and hypertension. And then all of a sudden you have more cortisol and insulin resistance. What condition does that make? It makes cardiometabolic disease, right? So right here in two SNPs that are co-inherited often together, add a little bit of stress and a little bit of salt into the environment, and we have cardiometabolic disease. Now let's take a little peek over here. What do we have here? We have sex hormone synthesis. So all of a sudden we start having erectile dysfunction and, oh, I'm sorry, honey, I don't want to have sex this year. Okay, this is where we start having all kinds of fertility issues and all kinds of other sorts of issues in the rest of our lives and in terms of our metabolic function. So these are things that we really need to be considering and looking at in terms of stress and mind-body balance, and looking in terms of optimal diet. So if you have a patient or a client who has issues with salt regulation and issues with stress balance, these are two pathways that you need to be concerned about. All right, biochemistry is over, I promise. Okay, so this is the study that talks about salt sensitivity in those two genes. And that angiotensin type 1 receptor, it does have a response for salt sensitivity, but only in certain populations, so specifically in Han Chinese. Okay. So next, more torturing with pretty foods. So again, I hope you're hungry with this beautiful salmon and avocado and eggs here. So we're going to talk about the paleo diet next. So this is eating as our ancestors did. So we're talking about wild-caught salmon. We're looking at fresh organic meats and eggs, fresh vegetables, nuts and seeds, higher fat but not very high fat. Some people will include ghee, clarified butter, those kinds of things. We're basically eliminating all of what we would consider our bad guys: dairy, gluten, grains, legumes, peanuts, refined oils, sugars, potatoes, processed foods, etc. When we're looking at the benefits of the paleo diet, it's helpful for autoimmune disease, type 2 diabetes, metabolic syndrome, acne, colorectal cancer, familial hypercholesterolemia, hypertension, cardiovascular disease. In most cases in clinical practice, we find this very helpful to transition people from the SAD diet or standard American diet into a healthier eating program. We do find that we have improved metabolic function, so better glycemic control, decreased A1C, decreased blood pressure, total cholesterol, triglycerides, BMI, etc. We do not find that it changes our intestinal permeability or cortisol levels, but it does show that and studies do support the fact that it does decrease C-reactive protein levels. This diet does have a 4-fold decrease in sodium and a 12.5 increase in potassium. So we just talked about this SNP, we just talked about ACE. So if we have patients that have those issues, then the paleo diet might be a really good option for those people. This diet also has considerably higher intakes of omega-3 fatty acids, particularly a 4-fold increase in omega-3 fatty acids. So if your patient happens to be a fish oil responder and happens to have a predisposition for high triglycerides, this diet may also be a really good choice for them. So this diet happens to have a 3-fold increase in fiber from all of the fruits and vegetables that they're consuming. So one caveat here is the fiber directly modulates the microbiome, but it also modulates our risk for colon cancer. So unfortunately, the microbiome is too big of a subject to talk about today, so we can talk about that next time. But we're going to talk about 2 genes that modulate our risk for cancer and diabetes. The first one is interleukin-10 or IL-10. And so what we find here is that the wild type carriers, even if they only consume 10 grams of fiber per day, they have a 27% reduction in their colon cancer risk. But if you have alterations in this gene and you don't consume at least 17 grams of fat, you're going to have a statistically increased risk for colon cancer. So what this suggests is that by modulating the fiber intake for those carriers, we can reduce their colon cancer risk. So this diet, specifically the paleo diet, might be a really good option if you find that your patients have alterations in this gene. So TCF7L2 is a gene that modulates risk for diabetes, and Dr. Hausman mentioned this gene to you guys earlier this morning. But there are two different SNPs in this gene that we're going to talk about. The first one, both of the risk alleles are T here, but the first one increases your risk for diabetes by about 67%. And this particular genotype has the traditional interventions for diabetes. So low glycemic load foods, reduce sugar consumption, limit grains. They should have a lower fat diet, increase their fiber, etc. But there's another SNP in the very same gene that basically contradicts this recommendation. So it's said that higher fiber intake may actually be protective only for non-carriers of this particular gene, and that higher fiber intake actually increased the A1C levels for those carriers of this gene. So basically saying that it's harmful for you to consume fiber if you have diabetes and carry this gene. So this is one of those things that makes you really take pause when looking at your concept of what is healthy and what you think of as the proper recommendation for diet and disease. Because when we start thinking about nutritional genomics and we start thinking about the actual actions that we're going to put forth for certain diseases, we really need to stop thinking about our concept of healthy. And when our patients come to us and say, well, I'm eating a healthy diet, like in Dr. Pisano's case study, she said, well, I'm eating a healthy diet. It may not be healthy for her. What may be perfect for one person may be poison for another, despite what their condition may be. And using our bio-individualized medicine is really what we need to be focused on. So other considerations for paleo, again, oxalate issues, very high in nuts and seeds. Protein, if your patient is coming to you for diabetes, one of our key aspects with diabetes is kidney damage. So make sure that you're looking for that. And then also, again, this diet is very high in fiber, so make sure that we don't have any issues with the microbiome or any sort of dysbiosis that may be exacerbated by high fiber. All right, standard American diet. And again, if you like burgers, this could be problematic. Hope you're not too hungry. All right, so high in saturated fat, high in processed meats, prepackaged foods, butter, fried foods, French fries, refined grains, potatoes, corn, high fructose corn syrup, sodas, results in all of our chronic illnesses today, diabetes, cardiovascular disease, cardiometabolic disease, etc. So what we really want to talk about here is when is a high-fat diet contraindicated? Because we all know that 90-year-old grandmother who eats a burger and smokes a pack of cigarettes a day and goes, I'm perfectly fine. So what we really need to drive home here is when is it really contraindicated? So when is it actually harmful to have a high-fat diet? So because we're at a nutrition conference, let's have a brief review of what these lipids actually are. So fats are stored as triglycerides and in adipose tissue, 1-glycerol, 3 fatty acids, thus triglycerides. Triglycerides typically increase with carbohydrate intake, not fat intake. And of course, they are our precursors to our sex hormones and cortisol, which we just talked about in the RAS system. So apolipoproteins, these are our big proteins that we're going to talk about today in terms of this. So they transport water soluble and fats soluble in one. Okay, the idea is that they're going to transport these things around the body, especially apolipoprotein E. They're found in the chylar microns and are intermediate density lipoproteins. They're really important in reverse cholesterol transport, breaking down these triglyceride-rich proteins, or fats, excuse me. So we want to make sure that they don't get stuck anywhere. We want to make sure that they move around the body freely and don't get stuck in our brains, don't get stuck in our arteries. All right, so we want to make sure that they're not altered and don't do anything they're not supposed to. So the big ones that we talk about are apolipoprotein E2, E3, and E4. These are our variants. Most of you are probably familiar with these. E4 variant is classically associated with Alzheimer's disease. Carrying one E4 variant increases your risk for Alzheimer's disease about 55%. Carrying two, about 90%. This, of course, is not our only causative risk factor for Alzheimer's disease. There are many others. But when we're looking at these apolipoproteins, again, their purpose is to transport these lipids around the body. When they get stuck and they don't work properly, we have disease. So we'll look about the genetics of this. What I really want you to focus on this slide is the population frequency. So we're looking at European ancestry, African Americans, and East Asians. So if you look at the E4, we're looking at about 3% of Europeans, 3.5% of African Americans, and only 1% of East Asians. This data is a little bit out of date. As you all know, our increased risk of Alzheimer's is increasing. But based on just this genotype alone, that would be our predictive odds ratios. Our neutral genotype, you'll see here, 57%, 42%, 75%. And then our E2 variant is our rarest genotype, so 0.3%, 0.4%, and 0.2%. So when we're looking at the algorithm of how to predict this, it's a little bit complicated, so I've got a cheat sheet up here for you when you get the lecture. The algorithm is a little bit challenging again, so just look at the cheat sheet. It'll give you the odds ratio. This is for Alzheimer's over here as well. This is the classic Yang study back in 2007 that started to associate E2 and E4 with cardiovascular disease and saturated fat intake. So the long story short here is that when you have increased saturated fat intake, you are going to increase your risk of cardiovascular disease and heart attack. So the take-home message here is that for those that have variations in those apolipoproteins, you should be on a low saturated fat diet. E2 is actually protective of Alzheimer's disease, which I mentioned, but it's associated with something called beta dyslipidemia. This is the patient that walks into your office and says, hey doc, I've got cholesterol and triglycerides in the 400s and it's been that way since I was 16, non-responsive to diet and exercise. This person should be on a very strict restriction of fat, typically saturated fat no more than 20 grams per day. Okay, so this is what you would call familial hypercholesterolemia or genetically high cholesterol. Most of the time, these patients do best on a vegan or vegetarian style diet. Another apolipoprotein is ApoA2. This one in diabetics is the gene that determines the requirements to prevent dyslipidemia. So currently the RDA says that less than 10% of your calories per day should come from saturated fats. Clinically, we find that this doesn't actually cut it for this gene. We need the kcals to be more about 1,500 and the ratio to be about 8%. This is an extreme fat restriction. So we're looking at about 6 grams of saturated fat per day, which is about 1 cube of cheddar cheese, guys. So we're looking at a very, very restricted fat intake for those that have the ApoA2 allele. So overall, when we're looking at these apolipoproteins, we're not talking about specific types of fats. Again, these are our fat carriers, so they don't care whether we're looking at the inflammatory fat or the not so inflammatory fats. They're trying to shuttle fat around your body. So the vegan diet is actually the best type of diet for the extreme genotypes, but remember, those are very, very rare genotypes. For the mixed genotypes, we need more information. When we're looking at the overall perspective in terms of the type of fat or the max fat for the genotypes, I've given you some recommendations here. But just to talk about the most extreme, so if you happen to have ApoA2 and you're an E2, you're looking at between 6 and 15 grams of fat per day. Okay, so really, really strict restrictions. All right, so now let's talk about peroxisomes. So peroxisomes, they break down very long fat fatty acids, and they do so by beta oxidation. So when we look at validation in nutritional genomics, we really want to look at our organic acids. So when you have peroxisome activation through mitochondrial function, you would expect to find elevations in things like adipate, suberate, and ethyl malonate. So if you're running organic acids tested frequently, you would know these things, you'll see these things. So when mitochondria are working correctly, the peroxisomes will break down these fatty acids and they will turn them into medium chain fatty acids and then have them turn into ATP. So we're really looking at energy source here. So they're also regulators of inflammation, and when they proliferate, they increase our fat burning ability. So this is really, really important when we're looking at whether or not we should consume fat or restrict fat. So these peroxisomes also control several genes that are associated with adipogenesis, lipid metabolism, and inflammation. So when you consume fat, if you have SNPs that work correctly, a high-fat diet will basically melt fat right off of your body, okay? So when you're looking at a high-fat diet or a ketogenic diet, if you have these SNPs down here, because I knew that would be a question, which SNPs are we talking about here? If you have minimal SNPs and peroxisomes, a high-fat diet is your friend. If you have errors in SNPs and peroxisomes and PPAR gamma, then a high-fat diet is not your friend, okay? It's going to increase inflammation and weight gain via insulin resistance. Okay, so these are things that you really need to be looking at because it inhibits some of our major anti-inflammatory pathways. So these are actually going to inhibit your ability to go into ketosis, and it's going to inhibit NF-kappa beta. So pretty big deal when we're looking at whether or not fat is your friend or your foe. All right, so fat mass and obesity SNP, our causative risk factor for obesity, requires alpha-ketoglutarate, oxygen, and iron. We basically need to put these people on a lower fat diet, also lower carbohydrate diet. We need to make sure they have adequate exercise because oxygen, molecular oxygen, we need to make sure they're breathing, make sure that those pathways are working correctly, support mitochondrial function. We don't need to tax their system with excess fat because the variant that is the causal variant here changes the way that their fat cells burn correctly. So, you know, when we're looking at a paleo-vegan style diet or a Mediterranean diet is often appropriate for these individuals. Fatty acid desaturase, also known as delta-6 desaturase. So if you're going to consider a higher fat diet, you want to have minimal SNPs in this particular gene. So if you've done NutraEvals, that's a Genova test, or if you've done fatty acid testing, this is one of the first genes in this pathway. But it's regulated by things like zinc and magnesium, B2, B3, vitamin C. It's also regulated by insulin. So if you have impairment in this pathway, it's going to upregulate your eicosanoids. Some of those are pro-inflammatory, some of those are anti-inflammatory, but most of the time if you have alterations in this gene, you're going to increase your thromboxanes and prostaglandins, which are not so friendly. Making some alterations, making sure that you don't have deficiencies in these cofactors that are listed here and controlling for insulin resistance can be very helpful with fatty acid desaturase. All right, peroxinase 1 or PON1. This is an esterase enzyme that helps to break down pesticides and pharmaceutical drugs. It's also involved in protecting HDL and LDL from oxidation. If there are errors in this gene, HDL becomes oxidized. So vitamin E and monounsaturated fats can be very helpful in protecting from this oxidative process. A high saturated fat all on its own, high saturated fat diet can decrease the activity of this enzyme. Okay, so if you just all on its own can be problematic for this enzyme. So the lactinase activity of this particular enzyme helps in the breakdown of homocysteine. So this is one of our connections when we look at our studies of how in the world does a high-fat diet increase homocysteine? This is how, okay? So when we start looking at that pathway that you learned about with Dr. Pisano and thinking, my gosh, how does diet intersect with this? This is one of the enzymes that does so, Okay, so if you're looking, I've got B6, I've got B12, I've got zinc, I've taken care of all of those things, glycine's fine, all of that stuff, and looking at that pathway and going, this person still has high homocysteine and maybe they're eating a high saturated fat diet, this could be why. Okay, look at their peroxidase enzymes. All right, last but not least is ketogenic diet. And I'm doing good on time. All right, so this is a very low carbohydrate diet. It's high in fat, and it's designed to put the body in a state of physiological ketosis. This is different from ketoacidosis. It typically contains meat and seafood, full fat dairy, and non-starchy vegetables. The fat ranges between 60 and 80% of caloric intake, and carbohydrates are very restricted at about 5%. One of the things that you should start thinking about, we've just learned about some saturated fat and fat restrictions and those kinds of things, and there are probably things that may be indicated or contraindicated for this diet. So when the body enters a state of ketosis, we're going to have a reduction in glucose supply, and the body is going to start using fats for energy. We're basically activating an alternative pathway in terms of energy metabolism. So ketone bodies are going to start being used by the central nervous system when we reach a concentration of about 4 millimoles. Normal levels are about 0.1 to 0.3 millimoles. And in normal glycemic individuals, we're typically not going to exceed concentrations of 7 to 8 millimoles. So let's talk about the new keto-adapted diet, so the new ways that we're using this diet today. So we're using it to treat heart disease, type 2 diabetes. We're using it for weight loss. We're using it for acne, cancer, Alzheimer's disease, Parkinson's disease, sleep disorder, increased athletic performance. And for many of these conditions, we're looking at increased mitochondrial function, and we're looking at lower blood insulin levels. So we're looking at these alternative pathways to try and activate the body in terms of the ketogenic diet. So of course, there's some considerations when we look at using something that is really extreme in terms of macronutrients. So there's some potential complications that we need to be aware of when we're medically inducing a diet. So we could have constipation, dysbiosis, indigestion, nausea, vomiting, hypoglycemia. We could have increased triglycerides, transient hyperuricemia, kidney stones, acidosis, growth disturbances, osteopenia. We could have nutritional deficiencies. We could have protein deficiency, magnesium deficiency, carnitine deficiency, sodium deficiency, iron deficiency, and selenium deficiency that could cause QT disturbances, which may cause cardiomyopathy. So this is something that really should be implemented by a trained professional, and these are things that we need to really be thoughtful of and be thoughtful of those things. So when we're looking at using we're looking at using the ketogenic diet in terms of epilepsy, in terms of a medically intervention diet, there are some times when the ketogenic diet is non-responsive. So when we're looking at that, there are some genes that actually determine the likelihood of there being a response to this, and so I've listed them here for you guys if you're using this in terms of epileptic intervention. So when is the ketogenic diet indicated? So peroxisomes, so don't forget about those. So if you have clear function in your peroxisomes, this diet is going to be incredibly successful for weight loss. So again, those peroxisomes control the expression of several genes associated with adipogenesis, lipid metabolism, and inflammation. And it's stimulated by fat intake. So if your peroxisome proliferation genes are clear, you are going to, again, have very, very high success with the ketogenic diet. Another gene, superoxide dismutase 1. So this is a transgenic mouse model of ALS, or Lou Gehrig's disease. And if you have errors in this particular gene, you're going to cause mitochondrial uncoupling. And so when we've studied this, it shows the ketogenic diet basically can regulate the mitochondrial function and ameliorate the symptoms of ALS. So this is incredibly promising when we're looking at complex diseases such as ALS. So one thing I want to caution you about, mitochondrial function is incredibly complicated. There are a lot of inborn errors of metabolism. There are a lot of genes that are in mitochondria that can glitch, that can have alterations. There are many people that when you try a ketogenic diet, a traditional ketogenic diet, not nutritional ketosis or keto adapted, but a traditional ketogenic diet, where you might actually have impairment in mitochondria through inflammation or through taxing beta oxidation. So if you are using nutritional genomics appropriately and also validating through testing at the same time, please be watchful of those things. Nutritional ketosis, on the other hand, can help to improve mitohormesis. But again, it needs to be done by a trained professional. So please be on the lookout for advanced training in the ketogenic diet. All right, and last but not least, when looking at the overall perspective of who could potentially be a candidate for the ketogenic diet, we're looking at our apolipoprotein gene again. So apolipoprotein E, this is the neutral genotype, so this would not have a fat restriction. So again, you're looking at about 57% of Europeans, about 42% of African Americans, and about 75% of East Asians would potentially be candidates for the ketogenic diet. So again, can't stress to you enough looking at your population when determining whether or not to implement on a certain nutrition or medical intervention. All right, so other considerations for the high-fat diets or keto-adapted diets. So high-fat diets alter unfavorably the Firmicutes to Bacteroidetes ratio and can disrupt the microbiome. There's so much about the microbiome, guys, that we just don't know about yet. So if you are implementing this diet and you notice that there are unfavorable changes, cease and realize that this is a whole big world that we just don't know enough about yet. Caution should really be advised for anybody who experiences anything that looks like a keto flu or an inflammatory cytokine storm. This is indicating significant pathway impairment. Anytime that you're starting to implement on something and someone has a negative reaction, stop, pause. You should say, I don't know enough yet. I need to dig deeper and figure out what's going on. Particle testing should be used on a regular basis. So not just your standard lipid profile, actually look at your particles. Make sure you're watching these things when you have someone either on a standard American diet or a higher fat diet, even in the paleo diet, and certainly on a ketogenic diet. Those things need to be watched and monitored, making sure that you're looking at those things. And last but not least, if you have someone who's not a candidate for something like the ketogenic diet, there's been a lot of research on intermittent fasting and modulation of the clock genes and the CRY1 genes, and they appear to have similar impacts on regulating insulin responses, and they may be a really good idea for those non-responders. So, in summary, so now that we've had a trip around the world in terms of diets, the majority of diets are medical diets, guys. These are therapeutic diets. So we think of diets and go, oh yeah, old hat. Okay, a lot of these diets have macronutrient ratios. They recommend a lot of nuts that might be contraindicated for someone who has genetic hyperoxaluria. They recommend a lot of fiber. That might be contraindicated from somebody. They recommend a lot of fish. That might not work for someone. When we start thinking about personalized precision nutrition, we need to start thinking about it in terms of actually having effect. Okay, so we look at the big population studies for Mediterranean diet. They have a measurable effect on overall mortality and wellness and longevity and Parkinson's disease and cancer and all of these things when we finally get 1.5 million people to participate in a study. So when we actually get participation, we find that diet actually works. Okay, we throw in nutritional genomics and precision medicine into that, then we really find that these things work and they work well. Okay, so when we look at things like that, we really start to have improvement and precision. So that's the point I want to make here. I did make you guys a pretty little chart in case you wanted to have it. So when you guys get the lecture, you'll have that there. There are a few extra little genes in here, but anyway, so Thank you, and I will turn it back over to Dana.